today we are going to solve Jan23 Biology Edexcel Unit 6. Question number 1. So here we are talking about an aquatic plant. They have given us some informations about these plants. They live in ponds, lakes, slow flowing rivers. They absorb nitrogens and grow rapidly, develops 3 leaflets, then divides to produce a daughter plant. That means the plant can have at max only 3 leaflets, not more than that. Okay, and in optimum conditions it takes 2 days to divide. That means in 2 days it will have 3 leaflets and then it will divide to produce a daughter plant. A. Describe an experiment to investigate the effect of nitrate ion concentration on the rate of growth of these plants. So here our dependent variable will be the number of plants or the mass of plants. As the plants will divide and produce daughter plants or develop new leaflets, the mass will increase. We will use 5 different concentrations of nitrate ions, and the plant will be set up with suitable apparatus, for example using beaker, because there will be enough space to count the number of plants separately, and we can use the same volume of each nitrate ion concentrations, and then we will count the number of leaves after 2 days or the number of plants, or we will measure the mass. We will control temperature and pH using water bath and pH using buffer, and we will have to keep the light intensity constant. And for the rate of to calculate the rate of growth, we have to use this formula, number of plants divided by time. And we are done with number 1A. B. Explain the roles of nitrogens in cell division to produce daughter plants. So nitrogen is used to form chlorophyll, amino acids, proteins for enzymes, to form RNA in DNA and ribosomes, and to produce tubulin involved in cell division. So here are the 3 points. Enzymes. For enzymes we are going to get 1 point. For chlorophyll we are going to get another point, and for amino acids and proteins we are going to get our 3rd point, and this is our extra point. And we are done with 1B. Number 2. Here we are talking about bacterial cells named E. coli. The growth of bacteria in a liquid culture can be investigated. Okay, the number of bacterial cells in the culture can be measured using the dilution plating technique. Identify one risk. Okay, this bacteria is pathogenic. So the risk is, while transferring the bacteria from one tube to another, we might get infected with it, as it's pathogenic, so it's harmful. How this risk could be reduced: incubate below body temperature, wear gloves, limit the lifting of lids. B. Here is a formula that we should memorize for exam, because This formula isn't given always in the question paper, so it's safe to memorize this formula. Already given for NT an initial number of bacteria. So after 2 hours we are getting 14,000 bacterial cells and the initial count was 900. So we will put the values according to the formula and we will get our answers. And I hope we are done with question number 1B. They mentioned the answer has to be in 3 significant figures, so 1.98 is the answer.